Hello, my name is Lena. I am a Bio 5A SI, and today we're going to talk about mutations and gene regulation. So let's start with mutations. So mutation is basically a heritable change in genetic material. So when we're talking about genetic material, it's mostly DNA, since that's mostly where we store our genetic information, such as proteins and enzymes. And in order to pass on mutations, it has to be in the gametes. It can't be in normal somatic cells like skin cells, muscle cells. It has to be in our gametes, so like our sperm and our egg. And what causes mutations is basically two things: either random chance, which is like spontaneous. So basically, our body is replicating DNA so fast, and there's so much DNA to replicate, so many base pairs, that there's bound to be at least one mistake once in a while. But sometimes it's not that big of a deal and it's normal. And the other kind is basically induced, where we throw so much damage to DNA and it has no ability to repair, considering how fast the damage is going, and that it just stays mutated and damaged. So examples could be X-rays, UV, or chemical mutagens. So any type of radiation that your body can't recover from because it's happening so fast. So an example that you could pull from real life is probably skin cancer from too much sun exposure, because the sun has lots of UV rays, and if we don't wear sunscreen, we get exposed to the UV rays, and that could cause genetic mutation. So there are different types of mutations depending on whether it's like on a global scale, like chromosomes, or once it's on the smaller scale, like base pairs. So and changes in chromosome number is a type of mutation. So like monosomy, trisomy, which is basically a change in aneuploidy. I think that's how you spell it. So an example could be trisomy 21. That's a common one. And other large scale mutations could be like inversions, translocations, or large deletions, especially in chromosomes. So like a change in the structure of a certain chromosome where things are found, or just a huge deletion of many genes in one chromosome. Those are types of like global scale mutations. And today we're mostly going to talk about point mutations, which is like changes in the small factors such as base pairs and codons. So there's two types. We'll first start with base substitution. So this doesn't change the number of bases in your genetic material, but it can also be altered. So there's three types. There's silent, missense, and nonsense. So silent, you could change one base pair, but it can still code for the same amino acid. So that's why it's called silent. You don't really even notice it because nothing really changed, but it's still a mutation. The second one is missense, which is one change in an amino acid could change the amino acid that you're coding for. And number three is nonsense, which is like truncation, where your change in amino acid could create a stop codon too early in your amino acid chain. Basically, the protein will never become, because you basically changed the genetic material and now it can never be what it originally was because it stopped too early. And the other type is insertions and deletions. So the main difference between this one is that this one could cause a frame shift and the number of base pairs changes. So compared to just one substitution where maybe one amino acid changed, like in missense, this one has a greater chance of changing many amino acids. So you can see an example here. Just by inserting or deleting one, you're changing the way that you're reading the genetic material. And so the codons change. Basically, what you originally wanted in your protein is probably not going to be the same as what you get with a mutation. It could be like halfway, but then all of a sudden it'll just change. So a case study is in hemoglobin and sickle cell disease. So what hemoglobin did is that it's basically a quaternary structure of a protein found in red blood cells that help carry oxygen to our tissues. And in sickle cell anemia, just one base pair substitution changed glutamic acid to valine. And this huge difference could potentially be deadly, where we can't carry enough oxygen to our tissues, and there could be like multiple underlying health issues. So it could affect all parts of the protein, from the primary all the way to the quaternary structure. And the reason it's so prominent in Africa is because basically, well, you first have to know about malaria. So malaria is a parasite that needs a host to survive, and basically it's from mosquitoes. So the mosquito carries its basically like egg that has malaria in it. It infects humans because malaria is basically a parasite, so it needs a host to survive. So it uses humans, and then it attacks our blood cells, and then it also basically reproduces, and the cycle keeps continuing. So when it has sickle cell disease, the parasite can't survive because it's not getting the specific nutrients that it needs specifically in our red blood cells due to the mutation. So if you are a carrier of sickle cell disease, the malaria parasite can't live in your body. And so basically, because of selective advantage and survival of the fittest, if you will, those who had malaria, those who had the sickle cell carrier gene would basically survive longer than those who either didn't have the malaria gene at all, they were homozygous, like homozygous no sickle cell, well, they would just die of malaria. And those who were homozygous for sickle cell anemia, they would just die due to underlying health conditions from having sickle cell anemia. So that's a specific case study that happened, and it's a pretty interesting phenomenon. So now let's go to gene regulation. So before we've talked about regulation of enzyme activity, and if you could recall back to like feedback inhibition, where basically you have a chain of enzymes that create a protein, and overall your end product could also inhibit the initial enzyme that created that whole chain, and that's how you basically regulate enzyme activity. So that same concept can be applied when we want to regulate genes specifically, like genes that code for those specific enzymes. So we could go even farther back to regulate protein and gene expression. So mostly for prokaryotes, we use something called the operon model, which is basically like we have these genes, all are functionally relatable to create specific enzymes, and they each have, and they're all connected, and they all have a promoter and an operator. So basically what happens is, without any sort of repressor on our operator, this gene could function normally, and it will create, transcribe, translate, all normal. But then once we realize that we've created enough, our co-repressor, which is usually our end product of this whole thing, can go inside a repressor, basically activate it, and that repressor will eventually suppress our operator, in which RNA polymerase can no longer basically transcribe our genes of interest. So that's basically how we regulate gene expression for usually in prokaryotes. But it gets much more complex once we get to eukaryotes, because we have like chromosomes and everything gets more complicated, because obviously eukaryotes are more complex cells. So we could go back just in a more global scale, just like we did with mutations. You could go on a global scale of gene regulation, like through chromosomes. So something basically called histone modification, where we know that chromosomes are basically wrapped around proteins called histones, and that's how we basically preserve them. So in acetylation, we're basically loosening that tightly bound structure of the chromosomes in order to promote transcription of genes on that chromosome. And methylation is basically the opposite, where we want to condense the structure even more because we want to prevent transcription of certain genes on that chromosome. And overall, DNA methylation is the same concept, except this is more of a long-term activation and can be passed down to multiple generations where this particular part of the chromosome is not activated at all. So this whole idea can be categorized into epigenetic inheritance. And as a quick side note, epigenetics is basically when we study genes, but we're not studying the basic changes in the genetic information or coding. It's more of changes that do not affect that at all. So there's no direct change in the DNA code, but you can affect other things like regulation. So for eukaryotic gene regulation, there's two basic concepts, two basic things that you want to keep an eye out for. Things called control elements, which can also be tied to upstream cis elements, and then transcription factors. So control elements are basically non-coding parts of the DNA that serve as binding sites for transcription factors. So if I had basically a gene here, basically we know that before the gene, upstream of it, we have something called a promoter. But before the promoter, there's also other things called upstream cis elements that basically help regulation of the gene. So before the gene of interest, which is why we call it upstream, it includes a Tata box. The Tata box is part of the promoter. And like general control elements, and then just distal control elements, which are just more farther upstream and farther away from our gene of interest. And they also include enhancers. And we also have transcription factors, which are basically certain proteins that bind to upstream cis elements to initiate or suppress transcription. So they could bind to the Tata box usually. And so you can either have an activator, which stimulates transcription, or repressor, which inhibits expression. So if you could recall from cell communication, basically that whole cascade and then our eventual product is a transcription factor, because overall what we want is to create proteins. And how do we create proteins? Through gene expression, where we want to transcribe a certain gene that we need depending on our situation. So basically our signal for that, our signal to either create and activate gene expression in order to get a certain protein depending on what's happening in our situation, we use transcription factors to help signal RNA polymerase that, hey, you need to activate this gene and we need to start transcribing it and translating it. So yeah, that's basically a tie-in to what we've learned before. And if you guys have any questions, feel free to leave it in the comments. And yeah, good luck on finals.